Introduction. Alright, let's begin our journal club discussion on the recent PNAS paper by XU and colleagues titled, "The transcription factor Bcl11a is essential for B1a cell maintenance during aging." 3, 4, 5. This paper reports that the transcription factor Bcl11a is a critical regulator for the survival of a specialized subset of innate immune cells, called B1a cells, as mice get older. 37. The authors show that Bcl11a expression is maintained at higher levels in aged female mice compared to males, which correlates with better preservation of their B1a cell numbers, and that Bcl11a achieves this by directly suppressing p53-mediated apoptosis. 11-13. Ultimately, the study positions Bcl11a as a key factor guarding these innate lymphocytes against the functional declines that accompany aging, particularly highlighting a mechanism for the known sex differences in immunity. 15. Paper categorization. So where does this paper fit in the landscape of scientific literature? I would categorize this work firmly as a new finding that is also good for teaching. It's a new finding because while Bcl11a's role in hematopoiesis and lymphocyte development was known 26, 27, 28, its specific, essential function in maintaining the B1a cell pool during the aging process and its connection to sex-dimorphic immune resilience had not been explored. 30. It's good for teaching because it provides a beautiful, self-contained story that integrates multiple levels of biological analysis, from organismal immunology and cell metabolism to transcriptomics and molecular genetics. It's a textbook example of how to mechanistically dissect a complex in vivo phenotype. Figure 1 sets the stage by characterizing the aging phenotype of peritoneal B1a cells. Panel A is just a workflow schematic. 194. Panels B and C show increased DNA damage, marked by gamma H2AX foci, in B1a cells from old mice, OM-OF, compared to young, YM-YF. 197. 198. Panels D and E demonstrate that aged B1a cells have higher rates of apoptosis, annexin V staining, and lower rates of proliferation, BRDU incorporation. 201. 202. Panels F and G are key. They quantify B1a cell numbers and proportions. They show that while numbers increase with age in both sexes, the expansion is dramatically larger in old females, OF, than in old males, OM. 213, 214. In fact, the proportion of B1a cells among all B cells drops in old males, but rises in old females. 214. Panels H and I measure mitochondrial membrane potential using a dye, DLC15, showing it's lower in aged cells, indicating mitochondrial dysfunction. 217, 218. Panels J through M assess cell metabolism using a seahorse analyzer. The key takeaway is in panel M, which measures glycoPER, or the glycolytic proton efflux rate. It shows that B1a cells from old females have significantly increased basal and compensatory glycolysis, while cells from old males do not, providing a metabolic explanation for their enhanced maintenance. 240. Okay, that's enough of the last figure. Now we're going to talk about the next figure, Figure 2. Figure 2 dives into the transcriptome and epigenome to find out why aged female B1a cells are different. Panels A and B are MA plots from bulk RNA-seq, showing differentially expressed genes. The key point is that old females, of versus yttrium fluoride, have far more transcriptional changes than old males, OM versus YM. 243, 244. Panels C and D are gene ontology, GO, analyses. Both sexes show changes in apoptosis and cell cycle pathways. 244. But uniquely, aged female cells upregulate genes involved in saccharide, sugar, metabolism, which fits perfectly with the glycolysis data from Figure 1. 397. Panel E is a heat map, visually confirming these changes in specific genes related to cell cycle, activation, apoptosis, and metabolism. 382. Panel F is a scatter plot comparing the fold changes between sexes, highlighting shared and distinct aging signatures. 383. Panel G is where it gets interesting. This is an ATAC-seq heat map, which measures chromatin accessibility. It shows a global increase in open chromatin in aged female B1a cells, but not in males, suggesting a major epigenetic shift. 401. Panels H and I pinpoint the potential cause. Panel H is a motif analysis of the newly accessible chromatin regions in old females, revealing that binding sites for the transcription factor Bcl11a are highly enriched. 405. 406. Panel I provides the direct evidence, a genome browser view showing that the Bcl11a gene locus itself becomes more accessible in aged female B1a cells. 407. Figure 3 validates the importance of Bcl11a in B1a cell maintenance. Panels A, B, and C confirm the findings from Figure 2. Using a Bcl11a EGFP reporter mouse, they show at the protein and mRNA level that Bcl11a expression goes up in aged females and down in aged males. 416, 417. Panel D is the first key loss-of-function experiment. Using MB1-CRE to delete Bcl11a in all B cells, they show a dramatic reduction in peritoneal B1a cell numbers in both young and old mice, of both sexes. 425. This demonstrates Bcl11a is required for their maintenance. Panels E, F, and G refine this experiment. To ensure the effect is intrinsic to B1a cells, they use a more specific, tamoxifen-inducible CRE driver, Brulhi41-CRE-RT2, which is expressed mainly in mature B1 cells. 431, 433. Deleting Bcl11a here also causes a sharp drop in B1a cell numbers, confirming its cell-intrinsic role. 434. Panels H, I, and J test the functional consequence. They use a cecal ligation and puncture (CLP) model, which induces polymicrobial sepsis. Mice with Bcl11 deleted in their B1a cells have fewer of these cells after CLP and, crucially, show significantly worse survival. 438, 439. This links Bcl11a's role in cell maintenance directly to protective immunity. Okay, that's enough of the last figure. Now we're going to talk about the next figure, Figure 4. Figure 4 investigates how Bcl11a maintains B1a cells, focusing on viability. Panels A through D show an in vitro competition assay. Bcl11a deleted B1a cells, CD45.2+, are co-cultured with wild-type WT-B1a cells, CD45.1+. The Bcl11a deleted cells are rapidly outcompeted and show much higher rates of apoptosis. 558, 559. Panels E through I move this experiment in vivo with an adoptive transfer. They mix Bcl11a deleted and WT-B-1a cells and transfer them into immunodeficient recipient mice. 562. Again, the Bcl11a deleted cells disappear over time, FG, which is associated with increased apoptosis, I, and decreased proliferation, H. 563, 566. The conclusion is clear. Bcl11a is essential for B1, a cell survival and fitness. Okay, that's enough of the last figure. Now we're going to talk about the next figure, Figure 5. Figure 5 drills down into the mechanism, testing the genetic interaction between Bcl11a and the master apoptosis regulator, P53. The hypothesis is that Bcl11a loss leads to P53 activation, triggering cell death. To test this, they created double knockout, DKO mouse, deleting both Bcl11a and P53 in B1a cells. 688. Panels A, B, and C show the steady-state result, while deleting Bcl11a alone decimates the B1a cell population. Deleting P53 at the same time significantly rescues the cell numbers. 689. Panels D, E, and F confirm this with another in vivo competition assay. They transfer B1a cells from the DKO mice, along with WT cells, into recipients. The DKO cells survive much better than the cells with only Bcl11a deleted, showing that P53 is indeed the downstream executioner of cell death in this context. 692. 693. Okay, that's enough of the last figure. Now we're going to talk about the next figure, Figure 6. Figure 6 aims to prove that Bcl11a directly regulates genes involved in the P53 pathway. Panel A is a schematic of a flag-tagged Bcl11, a mouse used for the next experiment. 869. The authors perform CUT and tag a technique to map where a transcription factor binds to the genome. Panel B shows the genomic distribution of Bcl11a binding sites, with a large fraction at promoter regions, 698. Panel C is a GO-analysis of genes whose promoters are bound by Bcl11a. The signal transduction by p53 class mediator pathway is significantly enriched, directly linking Bcl11a to p53 regulation, 699. Panel D provides the smoking gun. These are genome browser tracks showing strong Bcl11a binding peaks at the promoters of MCL1, an anti-apoptotic gene, and MDM2 and MDM4, which are famous negative regulators of p53, 787. Panel E confirms the functional outcome. Deleting Bcl11a leads to a significant decrease in the mRNA expression of MDM2, MDM4, and MCL1, 788. This closes the loop. Bcl11a binds to the promoters of these key survival genes and is required to maintain their expression. Okay, that's enough of the last figure. Now we're going to talk about the next figure, figure